Hi everybody, this is Dr. A with our second video on lab math. We're going to look at dilutions in this video. Okay, so quick review on proportions and ratios. This was from the previous video. Proportions are used to determine a quantity from a given ratio, and a ratio is an amount of something compared to an amount of something else. Ratios always describe a relative amount and at least two values are always involved. So in this ratio, for example, 1 to 9, what it could denote is 1 part of serum to 9 parts of a diluent like water or saline, okay, and or 1 part of whole blood to 9 parts of saline. And so that the parts, if you decide, for example, you do 1 meal to 9 meals, then you're using milliliters in both, okay? And so whenever you set up ratios and proportion problems, the two ratios being compared have to be written in the same order. So serum to diluent, that order, and they have to be in the same unit. So you cannot have milliliters on one side and microliters on another side of the ratio of the problem that you're trying to solve. All right, so let's talk a little bit about dilutions. It is often necessary to make dilutions of specimens that are being analyzed, or to make weaker solutions from stronger solutions. You are more likely actually to have to make a dilution of a specimen. This could be in clinical chemistry on analyzers if the initial reading is too high and out of the linearity or out of the measurement range of the analyzer, and you have to dilute it so that it can come back in the measurement range so it can actually be measured. Now, I will say a lot of the newer chemistry analyzers do a lot of dilutions by themselves. They do auto dilute, but they don't all do it, and some have only limited amount that they can do before it may fall on a lab tech to have to do dilutions. So a dilution is an expression of concentration, and it refers to the volume or number of parts of a substance to be diluted in the total volume or parts of the final solution. So for example, if you dilute something by half, you do a 1/2 dilution, you would take 1 part, let's say 1 meal of serum, and you would add 1 part or 1 meal of saline, for example. And so now you have 2 parts total, and it's a 1/2 dilution because you started with the 1 part or 1 meal of serum, and you're in 2 parts total. Specimens are often diluted using clinical lab reagent water, saline, a specific diluent, or level 1 calibrators. Always follow your protocol for the specific test that you need to dilute. And the dilution factor is always the bottom number of the fraction, and it is what you multiply the concentration of the diluted sample by to find out what the original concentration is. So in a sample, for example, let's say a glucose, if it was too high to read, and you dilute it by half, so you do a 1/2 dilution, 1 part of your serum with your high glucose and 1 part of saline or water, and you mix it and you run it again, and you get a reading of 350 milligrams per dl of glucose, then that was your 1/2 dilution. Well, the dilution factor is that bottom number of that fraction, so it's 2. And so what you do is you take that 350 and multiply it by your dilution factor, which is 2. So 350 times 2 equals 700 milligrams per dl. And so that was the initial concentration of glucose in the sample, which was too high for the analyzer to read, for example. If you express the dilution as a ratio, this specific one, it would have been 1 to 1 because you mixed 1 part of serum, the 1 meal of serum, to 1 part of saline, or 1 meal of saline. So when you write it as a ratio, it's what you're mixing. When you write it as a fraction, it's the initial part, what you're starting with, over the total volume, the total amount of parts. You can do single dilutions. They're, again, often done to get a very concentrated sample to read on an analyzer. You could do a 1/3 dilution or a 1/10 dilution, or you can even do a 1 to 100 dilution. Serial dilutions are a dilution series in which all dilutions, including or following the first one, are the same. They're often used to get antibody titer results. So, for example, you can start with a 1/2 dilution. We have 1 ml of your sample and 1 ml of your diluent, let's say water. Okay, so that's so 1/2, and then you take 1 ml of your 1/2 dilution, put it in the next tube, and add 1 ml of water to that, and so that becomes a 1/4. So it's the 1/2 of 1/2. And then you can take 1 ml of your 1/4 and add another ml, and put it in a different tube, add 1 ml of water or saline to it, and now you have a 1/8. And you just keep going, diluting it by half, by half, by half, by half, all the way down, and you can go quite far with that. You can also do it by 10, so you'd be at 1/10, then it'll be 1/100, 1/1000, and so on. So a 1/2 dilution is made by mixing 1 part of sample with 1 part of diluent, so it's that 1-to-1 ratio for a total volume of 2 parts. For example, 1 ml of serum plus 1 ml of water for a total volume of 2 ml, just making it a 1/2 dilution, and the dilution factor is 2, which is what you would multiply the result back by, or what you would program in the analyzer if you're trying to program the dilution. A 1/3 dilution, for example, is made by mixing 1 part of sample with 2 parts of diluent, so as a ratio is 1-to-2, for a total volume of 3 parts, or a 1/3 dilution. For example, you would have 1 ml of serum is your 1 part of serum here, and 2 ml of water. There you go, for a total of 3 ml, thus making it 1/3, and a dilution factor is 3. A 1/10 dilution is made by mixing 1 part of sample with 9 parts of diluent, so it's a 1-to-9 if you write it as a ratio. The total volume is 10 parts, so again, if you had 1 ml of serum, 9 ml of water, so total of 10 ml, so it's a 1/10 dilution, and the dilution factor then is 10. That's what you would multiply back the results back by to get your initial concentration. So an example problem would be a blood glucose is reading too high for the analyzer to properly read. The lab tech makes a 1/2 dilution by mixing 100 microliter of the plasma with 100 microliter of clinical lab reagent water, and runs the sample again. The machine gives a reading of 526 milligrams per dl on the diluted sample. The original glucose concentration in the patient's plasma is going to be 526 milligrams per dl times 2, which is the dilution factor, so it's 1,052 milligrams per deciliter. So that's just one example problem. There are so many problems that you can have and solve, but just have to think your way through it. I have other videos on dilutions on the YouTube channel that you can check out, and if you have any questions in particular, just please let me know.